Hello, and welcome to this e-seminar which will provide an introduction to animal and dairy-derived dietary protein substitutes. Please note that the views, thoughts and opinions expressed during this presentation are those of the author and do not necessarily represent the views of the UK Government or its associated departments. This e-seminar provides a brief introduction to alternative dietary protein sources to those from meat and dairy derivatives, hereafter referred to as alternative proteins. The background surrounding the reasons for exploring animal and dairy-derived protein substitutes will be described, and a synopsis surrounding alternatives currently being explored will be presented. Limitations relating to each alternative, as well as any health and safety concerns, will be discussed before a brief summary of the e-seminar is presented. Current global food production practices are reported to be associated with the production of high levels of greenhouse gas emissions, such as methane and sulfur dioxide. In a recent report by the UK Committee on Climate Change, it was suggested that at least a 20% reduction in the consumption of animal and dairy products would be required in order for the UK to achieve net zero carbon emissions by 2050. The report also indicated that switching to sustainable, alternative dietary protein sources may be one strategy that would help reduce the impact of food production practices on the climate. However, this would require improving the UK's current level of protein self-sufficiency and increasing the diversity in sources of protein, as well as diversity of end-user products. There is now a growing interest in identifying alternative sources of dietary proteins to help reduce the demand for meat and dairy-derived sources. It is recognised that numerous nutritious alternative protein sources already exist, such as those derived from plants and seaweeds, or those produced via non-animal cell culture or fermentation of fungi and microalgae. More recently, techniques for producing laboratory-cultured meat have been suggested as being a viable approach to traditional agricultural methods. Collectively, these alternative sources of dietary proteins may offer the potential to provide safe, affordable, and sustainable protein sources for the growing global population. It is not currently anticipated that these alternative proteins would entirely replace meat and dairy-derived proteins, but would help alleviate the growing global demand and supplement the intake of communities where essential dietary protein consumption is below that required for good health. Non-meat and dairy-derived sources of protein are not a new or recent concept. The food industry already caters for vegetarian, vegan, and specialty diets. Because of this, an extensive market in alternative protein materials already exists, which includes ingredients that have been extensively reprocessed to closely resemble meat and seafood products in taste, appearance, and texture. As well as emerging and novel dietary protein sources, established products such as tofu and mycoprotein are also regularly employed as ingredients in traditional meals as replacements for conventional animal-derived protein. However, industry is now targeting a broader market of consumers who seek to reduce their meat consumption rather than avoid it. Hence, the strategies industry has adopted include the manufacturing of products which mimic the varied tastes and textures of traditional meats and seafood. Adoption of alternative proteins is currently driven by consumer preference rather than governmental short- and long-term goals. Consumer and manufacturer surveys have generally indicated that the primary drivers that regulate product adoption tend to be cost, apparent sustainability and eco-friendly production, health implications, and product nutritional content. While primary issues for concern include genuine product sustainability, potential health risks from possible allergenicity issues, as well as those originating from the production process employed, for example the use of antibiotics with cell culture-based approaches, and from the risk associated with long-term consumption, for example high levels of salt, sugar and fats. And finally, the product's nutritional profile, for example the amino acid composition. In terms of economics, cost of production as well as retail price. Level of consumer acceptance, including taste and palatability, have both been recognized to significantly influence product uptake by consumers. The groupings for sources of alternative dietary proteins shown on the slide reflect those employed in the current scientific literature and have generally been adopted by the food industry. These include plant-based meat substitutes, which are products based on protein extracts from plant material and processed to achieve meat-like tastes and textures. Novel protein sources, which include non-traditional sources of protein such as insects, seaweeds, and microalgae. Notably, many novel foods to the UK and Western markets are not novel to other cultures and are employed to provide a regular source of dietary protein by a number of cultures around the world. Precision fermentation-based meat substitutes. These proteins are ingredients generated through the use of precision fermentation and biomass fermentation, either in enclosed culture vessels or open ponds. And finally, cultured meat protein. This source of protein is the more recent and refers to in vitro, lab-grown, or synthetic meat and is comprised of primarily animal muscle cells grown under laboratory conditions. Plant-based proteins represent one of the oldest substitutes for meat and dairy-derived protein and include tofu, soybean curd, tempeh, fermented soybean, and yumba, dried soybean curd. Plant-based proteins are also the current leading segment of the alternative protein market. The primary sources for plant-based protein include those derived from soybean, legume, oilseed, wheat, rye, and barley. The dietary consumption of these proteins is not equivalent to conventional plant and animal-based diets. Although they are promoted as a healthy, environmentally friendly alternative source of dietary protein, and so share many of the benefits associated with the adoption of a vegetarian or vegan diet. However, the perception that these products are healthier for the consumer is not entirely true, as they are generally ultra-processed foods manufactured to mimic protein content, appearance, taste and texture to that of conventional animal-derived protein sources. In terms of benefits, plant-based alternative proteins are currently promoted as eco-friendly, sustainable, nutritious and healthy sources of dietary protein. Nutritionally, they have been reported as a good source of protein, with a content in the general range of 10 to 20% dry weight for dry weight. However, protein quality in many cases may be of only limited nutritional value due to the frequent lack of essential amino acids such as lysine, methionine, and cysteine. In general terms, they are frequently employed in the forms of whole seeds, powder, flour, or as protein isolates, extracts, with different sources of plant protein being more suited to different applications, dependent upon their physical properties and nutritional composition. For example, soybeans or soy protein have been used as meat extenders and meat analogues, a role that's commonly addressed with the use of textured vegetable protein, or TVP, as it is commonly referred to. A variety of alternative sources of dietary proteins offer a high protein content compared to conventional meat and dairy sources. Some of these may be considered as novel food types, but are not always new to humanity, and may have long been part of the diet in many cultures. Examples of these novel sources include edible insects, microalgae, and cyanobacteria, microalgae, which include kelps and seaweeds, edible jellyfish, and filamentous fungi. However, production of these sources at a realistic industrial scale often involves novel production technologies and exposure of the products to man-made environments and processes which may introduce a range of unforeseen problems. There are currently 2,000 species of insect that are regularly consumed in 113 countries, primarily in Africa, South America, and Southeast Asia. In terms of commercial production for Western markets, crickets, black soldier fly, grasshoppers, and mealworms are the insects of current interest due to their high nutritional values. They are reported to be good sources of fats and proteins and provide adequate levels of vitamins and essential amino acids. For Western markets, they have primarily been marketed as ingredients for use in processed foods, the market categories of which include animal and pet feed, processed insect powder, and baked products and snacks. In terms of benefits, insect-derived protein provide a high feed-to-meat conversion ratio while offering a lower land, water and feed requirement. Incorporation into human foods or animal feeds follows a similar approach to that employed with plant-based protein, in that it is used primarily as an ingredient in highly processed products. However, insects are potentially a more nutritious source due to a higher protein, fatty acids, mineral, and vitamin B content. Examples of frequently consumed species include species of leafhopper, planthopper, and cicadas, as well as beetles, termites, crickets, and locusts. Insect protein content has been reported to generally vary between 13 to 81% dry weight, and examples of the nutrition content including protein of regularly consumed insects is summarized in the table shown on the slide. However, nutritional value is strongly dependent on species, sex, and metamorphosis state. In terms of protein quality, insect protein generally exhibits both a high digestibility index score and good essential amino acids profile. In terms of limitations, consumer skepticism to novel insect-derived foods is currently the largest hurdle to be overcome. However, there have also been a number of health and safety-related risks linked to the consumption of insects, primarily attributable to rare occurrences of consumer allergenicity to insect exoskeleton chitin, but also to potential microbial and heavy metal contamination, as insects have the capability to accumulate these types of contaminants from their feed sources. As a food in its own right, the Asian markets tend to cater for insects that have been cleaned, cooked and served whole. However, with respect to the EU and UK, insect-based ingredients may currently be the best means to facilitate their adoption as a food resource. Drying and milling approaches have been widely employed in this respect, and insect powder could be added as ingredients to foods such as bread, biscuits and sausages to enhance their nutritional content. Protein and oil processing methods have also been evaluated as a means for extracting insect-derived protein and oils. The term macroalgae is used to refer to all of the species of seaweeds and kelp, and in those communities around the world that regularly consume them, they are generally thought of as a sea vegetable. Many of the coastal communities in Europe use them as food, animal feed and fertilizer, and an increasing number of commercial applications are now targeting algae biomass utilization. At the time of compiling this e-seminar, the European Joint Research Council Algal Database indicated that there were 413 enterprises within the EU and other European countries who were active in the production of macroalgal biomass, which is harvested both from wild stocks and aquaculture systems. In terms of stable yield, aquaculture has been demonstrated to provide a more stable environment for biomass production, although harvesting from both the sea and ocean still provides a major contribution to annual production volumes. The largest established industrial market is currently located in Asia, where an annual production in the region of 32 million tons of aquatic algae has been reported. Seaweed species commonly consumed in the UK include pepperduls, gutweed, Dumont's tubular weed, laver, also known as nori, carrageen, false carrageen, dulse, sea spaghetti, sea lettuce, and bladderwrack. The Latin names for each of the examples provided is also shown on the slide. In terms of benefits, macroalgae are easily cultivated and may be grown in freshwater or saltwater environments. They can provide for a nutritious food source, are generally high in dietary fibers, vitamins and minerals while being low in dietary fat. Seaweed can also contain high amounts of potassium, which may be harmful to individuals with kidney disease, as well as vitamin K, which may interfere with blood-thinning medications such as warfarin. Some varieties of seaweed may also accumulate high levels of heavy metals such as arsenic, cadmium and lead, as well as iodine. Macroalgal species have been reported to be generally good sources of protein, with some species possessing up to 50% protein by dry weight. However, seaweed protein content differs significantly between species, with brown species typically having a protein content of 3 plus or minus 15% by dry weight, and green or red seaweeds generally exhibiting a protein content in the region of 10% plus or minus 47%. This figure can vary greatly by geographical location, stage of maturity, and season of harvest. In terms of composition, amino acid content has been reported to be comparable to protein sources such as eggs or soybean. Macroalgae represent not only a source of alternative dietary protein, but potentially also a source of nutritional and functional food supplements. They also offer chemical-free fertilizers and the potential to be incorporated into aquaculture diets, fish feed, or animal feeds. Jellyfish are primarily marketed and consumed in China, Japan, and South Korea. End-of-year estimates for 2018 recorded the level of global production at around 300,000 tons. Jellyfish are primarily harvested in wild fisheries, although examples of large-scale aquaculture are in the early stages of operation. Harvested jellyfish are typically processed through a multi-phase drying process over a number of weeks into a semi-dried and/or salt-preserved product. Desalted, ready-to-use jellyfish are low in calories and fat and are typically reconstituted through soaking in water prior to use. They are a good source of essential macro and micro elements, including proteins, fats, particularly omega-3 and omega-6 fatty acids, antioxidants, polyphenols, and minerals, e.g. iron, selenium, calcium, magnesium, and phosphorus. In general, they lack flavor and are used to help add texture to a variety of culinary dishes. In terms of protein content, this is generally collagen-based and may be as high as 50% by dried weight. As well as a food, the protein content may also serve as a source of bioactive compounds following enzymatic digestion for use with pharmaceutical and biotechnological applications. As highlighted on the previous slide, jellyfish are primarily harvested in wild fisheries, although large-scale aquaculture operations initiated during the 1980s. Global production has been estimated at around 10,000 to 17,000 tonnes a year between 2011 and 2015, with the harvest generally being processed into semi-dried products using multi-phase drying processes. The dried product is typically employed as a food ingredient or as snacks, although other uses may extend to use as a food in its own right or in animal feed. They are also a potential source of bioactive compounds for the pharmaceutical and biotechnological industry. In terms of health and safety considerations, there are no reported physical risks associated with the general consumption of jellyfish. They do possess a number of toxic substances in their stinging appendages, which may cause serious injury if not removed or cooked correctly. They have not been indicated as a major risk with respect to microbial contaminants or for allergenicity issues. There have been infrequent incidents of IgE-mediated food hypersensitivity to jellyfish, but at the time of compiling the e-seminar, these are comparatively uncommon. Jellyfish are also susceptible to bioaccumulation of chemicals and heavy metals, for which regular screening is recommended. Commonly consumed edible species of jellyfish include cannonball jellyfish, jelly blubber jellyfish, moon jellyfish, and Nomura's jellyfish, examples of which are shown on the slide. Precision fermentation, or culture, is the term used to refer to the industrial process of growing a range of microorganisms in large-scale culture systems. Those organisms regularly cultured include species of yeasts, microalgae, bacteria, and fungi. Microbial protein, along with a variety of substances, is generally isolated from the dried cells of microorganisms and added as an ingredient to food or feeds to enhance their nutritional content. As an approach, it offers the opportunity to maximize the use of valuable resources such as land, water, and nutrients. The process of large-scale culture of microbes is a well-characterized process and dates back to the production of various beverages such as beer and wine. More recently, the approach has been employed with the production of products derived from microalgal and fungal culture. Scaling of the process to industrial proportions is relatively straightforward, and as such it has become a focus for increasing the diversity of protein source options for a variety of consumers. A key limitation of precision fermentation approaches is that the culture conditions employed may make microbial biomass production susceptible to contamination from a range of substances, but particularly heavy metals, pesticides, and PCBs where wastewater sources are employed. In addition, accumulation of biotoxins may also be problematic where the culture environment becomes contaminated with toxin-producing microbes, such as the cyanobacterium microcystis aeruginosa. A further consideration, and that currently the subject of a comprehensive safety review, is the additional hazard posed by potential allergens. In terms of protein content, ranges in the region of 27 to 64% dry weight have been reported in the scientific literature, although culture biomass may be used as a whole, e.g. spray-dried, or alternatively the protein extracted from this. Common sources of fermentable microbes used for food businesses are microalgae and mycoprotein. The successful Quorn brand of vegetarian products is itself the result of the precision fermentation of the filamentous fungi. Fusarium venenatum and various microalgal-derived products have been used in beverages such as shakes and smoothies. Microalgae are a diverse group of microorganisms estimated to be comprised of approximately 200,000 species. Industrial-scale production cultivation evaluations were begun in the 1980s and initially focused on spirulina and coleria species. Currently, and as a source of microbial-derived proteins, Dunaliella salina, Haematococcus pluvialis, and Phaeodactylum tricornutum are more commonly utilized by the food industry, the outputs of which are primarily sold on the Asian market in the form of dried algae. Nutritionally, they are a good source of proteins and carbohydrates, may be used as ingredients in a wide range of products, and have been successfully employed in a number of food supplements. In terms of benefits, microalgae have the potential to manufacture proteins more efficiently than terrestrial plants, exhibit carbon dioxide fixation rates that are on average 5 to 8 times higher than terrestrial plants, and are able to use different forms of nitrogen fertilizer, e.g. nitrate, nitrite, ammonium, etc. Their cultivation is possible in both open ponds or enclosed bioreactors, the choice of which determines what nutritional inputs are required. With open pond systems, phototropic-driven production predominates, with primary inputs being sunlight for photosynthesis, atmospheric CO2, and nutrients from the environment. However, with enclosed bioreactor systems, heterotrophic production predominates and cultivation is undertaken in fermenters using a carbon-rich culture media. Microalgae are currently exploited for both biological and industrial applications, including production of human, animal, and aquaculture feeds, production of functional ingredients, pharmaceuticals, abrasives, through dietary culture, and also biofuels. Fungi have become the subject of increasing attention as a low-calorie source of protein, as they provide a texture suitable for adoption with meat and seafood substitutes. In addition, they require less land, energy, and water for growth than comparable arable systems. Fungi are not a novel protein source to Western markets. Fusarium venenatum has been cultured as a source of mycoprotein on an industrial scale since the 1980s for use in the production of Mondi Nissin's vegetarian corn products. More recently, a related Fusarium species, Fusarium flavolapis, has been employed by Nature's Find for the production of mycoprotein for incorporation into their vegetarian Phi product range. Mycoprotein yields from culture are typically high, and the product can be formed into a range of shapes through the use of a combination of heat and pressure. The industrial scale culture of fungal species also has potential for use with genetically manipulated organism (GMO) technologies, particularly in the production of non-fungal proteinaceous substances, such as bovine albumin or casein. In terms of benefits, mycoprotein provides a texture suitable for adoption with meat and seafood substitutes. Its production requires less land, energy, and water than comparable arable systems. The organisms used in its production have the potential for use with genetically manipulated organism (GMO) technologies for non-fungal protein production. Unfortunately, the Fusarium species currently employed for its production require glucose-rich culture media that is supplemented with micronutrients. One possible solution may be the adoption of agri-food waste as a nutrient source, but the potential downstream implications for this, particularly bioaccumulation of heavy metal and chemical contaminants, could be problematic. In addition, the current culture conditions required with the growth of Fusarium sp leads to the generation of potentially high concentrations of RNA in culture, 1% weight to weight, requiring inclusion of a routine reduction step. In terms of macronutrients per 100 grams dry weight for corn mycoprotein products, analysis indicates levels of 45% protein, 25% fiber, 13% fat, and 10% carbohydrate. However, product protein content may be higher due to the routine inclusion of egg proteins, which may be added to assist with texturizing the final product. Fungal mycelium may also be used to provide an alternative for scaffolding during cell culturing for cultured meat, fish, and fat processes. Cultured meat, also known as in vitro, lab-grown, or cell-based meat, is produced from animal cells that have been multiplied through culture in controlled settings, and which utilizes tissue engineering techniques that were pioneered in the field of human regenerative medicine. The concept was first patented for use with generating fish skeletal muscle in the 1990s, with the first proof of concept demonstration undertaken in 2013. This demonstration from Professor Paul Post of the University Maastricht involved the generation of a hamburger patty using cultured animal muscle stem cells, but at an unrealistic equivalent retail cost of $280,400 per burger. However, rapid advances in the field have recently resulted in the commercial sale of cell-cultured meat products at a more realistic retail price, with customers potentially expecting to pay in the region of $10 a burger. In terms of benefits, cultured meat may be expected to have all the attributes of conventional meat products and thereby satisfy the consumer's nutritional and sensory requirements. It may also offer the potential to reduce environmental impacts and aspects of climate change through addressing some of the environmental issues associated with meat production, such as greenhouse gas emissions. It would also address the issues of animal welfare, food security, and aspects of human health. In terms of limitations, a key concern surrounds the ethical considerations that are associated with the culture conditions employed. Conventional cell culture approaches employ animal-derived components, including large volumes of blood serum. However, technological developments are leading to the development of serum-free media, but adoption of this will require additional culture condition optimization. In addition, scaling of the different components of the process for mass production are still being explored. For example, culture vessel design, sometimes referred to as bioreactors, still will require refinement. Reports in the relevant literature also indicate that different bioreactor types may significantly influence mass production capabilities, and that culture efficiencies may vary between cell lines, which infers that culture conditions will require evaluation on a case-by-case basis. Finally, current high production costs mean that the approach currently only addresses a very niche market. In terms of expected protein yields, this remains unspecified, as the approach is still in the development stage, and the use of mixes of cell lines as well as extracellular matrices to help provide product form have still to be fully determined. As the products are produced under sterile cell culture conditions, health and safety concerns are limited, as in theory there should be no unwanted chemical or microbial contamination present, provided the process is managed appropriately. Similarly, hypersensitivity or allergenicity issues should not be a problem, as inputs to the process will be strictly controlled. The development and acceptance of non-livestock-derived dietary protein has primarily been driven by a combination of issues, including the need to preserve land and water resources, addressing animal welfare concerns, and the pursuit of environmental sustainability. Current examples of sources for alternative dietary protein include plant-based meat substitutes, novel proteins, including edible insects, seaweed, and edible jellyfish, precision fermentation products, microalgae, and mycoprotein, and potentially cultured meats. With some of these already available to the retail consumer, including textured wheat and soybean proteins, which have been available to the consumer for some time, edible insects, which are already added to some foods to enhance their nutritional value and of quality, and precision fermented fungal biomass, such as that available through the Quorn TM brand. However, global food production practices are associated with the emission of high levels of greenhouse gases, which contribute to global warming and climate change. In particular, the agricultural rearing of livestock generates a significant proportion of these emissions. As indicated in a report by the UK Committee on Climate Change, switching to sustainable alternative dietary protein sources may aid in the reduction of these greenhouse gas emissions, while also aiding in addressing other critical environmental issues such as land and water usage. However, if alternative protein sources are to make a significant contribution to reductions in emissions, considerable progress in the field will be required, including further technical developments such as the optimization of culture conditions for in vitro meat production, consumer acceptance of some novel foods and their products, and the formulation of guidance and regulations. As an added incentive to finding solutions to these hurdles, the UK Committee on Climate Change report referred to earlier in this presentation indicated that a 20% reduction in consumption of livestock-derived protein products would be required for the UK to achieve the target of net zero carbon emissions by 2050. This e-seminar has been produced as part of the Joint Knowledge Transfer Framework for Food Standards and Food Safety Analysis, which is funded by the Department for Environment, Food and Rural Affairs, Defra, the Food Standards Agency, FSA, Food Standards Scotland, FSS, and the Department for Science, Innovation and Technology, DSIT, via the Government Chemist Programme 2020 to 2023, and is aimed at disseminating knowledge from government-funded research to stakeholders to support UK laboratory capability and promote best practice in food safety and standards analysis.